If I'm not, Kelsey, you let me know. So today's class is on viral vaccines. We are not essentially going to speak about only viral vaccines. We might mention 1 or 2 or 3 bacterial vaccines as we proceed. So the title is viral vaccines, but you might see some bacterial vaccines when you're giving examples ahead, so don't worry about that. So when we talk about vaccines, there are a lot of stories surrounding vaccines. And what we have to appreciate is that vaccines have been very integral part of the healthcare system. With vaccines, we've been able to prevent some deadly diseases that previously caused a lot of infections and death in people. We've been able to, eradicate some diseases. And we've also been, as from the previous years, with the COVID-19 era, we've been able to see how vaccines have become very important. So it will be upon each and every person to appreciate or to tell whether they think vaccines are important in the day-to-day health care activities or not, because we also have people who have controversial issues around vaccines. So depending on what your take is as a healthcare person, it will all come into your own judgment at the end of the day. So just to give an outline of this presentation today, we'll be able to learn about some terms that surrounds vaccinology, give a little bit brief of the history of vaccines, some routes of administration of the vaccine, a little bit on the principle of vaccinology, and this goes back to our basic immunology. If we are able to understand some basic immunology, then we will be able to know what exactly vaccinology is. We'll look at some types of vaccines and how they are prepared, advantages and disadvantages, and slightly touch a little bit on some recent advances in vaccinology. So just to define, a vaccine is an immunobiological substance that usually resembles a pathogen, and it produces an acquired immunity to a particular disease. So it's something that is introduced to the body, and the body sees it as foreign, and it resembles a pathogen that the body that normally will see as foreign, and the body mounts an immune response against it. Well, on the other hand, vaccination is the act of administering this particular, whether it's either an antigen, a part of the pathogen, or the whole pathogen, so that the body can now produce an immunity against that particular pathogen. And immunization is the process by now whereby the body gets immunity to that particular pathogen. So it's an entire process that is also categorized on its own. We can see, we'll be able to see how immunization can be acquired, and vaccination is one of the methods that you can use to immunize people. And an adjuvant is just a pharmacological or immunological agent that will modify the effect of a drug or a vaccine. So the main function of an adjuvant in either vaccine or a drug is to enhance the immune response to a particular agent when it's introduced into the body. So it helps to mount a more huge immune response. And then an example is like the aluminum salt. So just to give a brief history of vaccines, vaccines, there are some practices that people used to do earlier back in the years that gave us gave us a foundation on vaccinology. And some of these included, you see, like the Buddhists drinking the venom, the snake venom, to confer immunity to this when they're to the snake venom when they're bitten by the snake, or something that most of us know is when Edward Jenner was able to, inoculate an 11-month-old boy with vaccinia virus to prevent smallpox. So during this period, Edward Jenner was able to identify that those maids who used to milk cows, they would get lesions on their hands, but they would not fall sick from those lesions. So that he was able to take some of the go to the lesions on the hands of the maids, get some of those fluids from there, and that is what he used to inject the 11th month baby. And later, when the baby was injected or was exposed to vaccinia virus, the baby was not able to get smallpox. So that was he now was considered as the father of immunology, in that that was like the basis or the foundation for vaccinology. You can read more about the history of vaccines and find more interesting stories on the same. Yeah, if you really want to. So what are some of the routes of administration of vaccines? So vaccines can either be injected, they can be taken orally, they can be taken through aerosols. Most of the time it's intranasal. So just to give some examples, the oral vaccines include the OPV, that is the Sabin, that is given for polio, and the rotavirus. So this is giving just a little bit of examples. You can read further on the same. And the intramuscular, I said we will not only focus on viruses, because some of these, like the DTaP, is for bacteria. The TT, that is the tetanus, is for bacteria. But if you want to concentrate on viruses, we have hepatitis B that is injected intramuscularly, and also IPV, that is the Salk for polio vaccine. So you can go through those examples at your own time. An example of vaccine that is injected through the subcutaneous route includes the measles and the yellow fever, and the intradermal includes the BCG for TB. So this is a picture that is giving an example of intranasal route, and this is usually mostly for the influenza, the flu shot. So at the end of the day, we need to understand how these vaccines work, how they prime our immune system, and we need to also know that when we are taking a vaccine, the ideal vaccine is that vaccine that will ideally give immunologic memory. So we'll have memory B cells, memory T cells, T helper cells, and memory cytotoxic T cells. And also at the same time, an ideal vaccine is one that will give minimal side effects, and it will not give an infection, in that it is safe to take. So that is what an ideal vaccine is. It gives immunological memory, which cuts across both humoral and cell-mediated, and also is safe, meaning once the vaccine is introduced into the body, there are very minimal chances of someone getting an infection from that vaccine. And at the same time, the side effects are also very minimal. So as we go ahead, we will see that not all types of vaccines will be able to do this. So some will give memory on the humoral arm. Some will give both humoral and cell-mediated memory. And so depending on what is needed at what time, then a vaccine can be chosen. So from this principle of vaccinology, we'll just take an example of a vaccine that elicits both humoral and the cell-mediated arm. And ideally, that will be a vaccine that is a live attenuated vaccine. So for example, if someone is given a vaccine that is a live attenuated vaccine, we have our antigen presenting cells, an example, which is the dendritic cell. So our antigen presenting cells from our immunology, we all know that they express both MHC class 1 and MHC class 2. This is MHC class 1, and this is MHC class 1, sorry, and this is MHC class 1 and 2, sorry. So our APCs, they present both class 1 and class 2. And class 1 is presented in all nucleated cells, but our antigen presenting cells will have both class 2 and class 1. So once an antigen is injected, this antigen presenting cell, which is an example, a dendritic cell, will either uptake this antigen through phagocytosis and then present it on MHC class 2. So once it presents it on MHC class 2, it's presenting it to the CD4 helper T cells. So now this is taking the arm of the humoral. We are discussing the humoral arm, then we will come back and discuss the cell-mediated arm. So once it presents this antigen, which are the peptides of the viral peptides on MHC class 2, it presents it to the CD4 helper cell. So the CD4 helper T cell produces a lot of interleukins. You can check them out, the kind of interleukins that are produced by T helper cells that will go ahead and lead to the proliferation and maturation of the B cell. So that once the B cell matures and proliferates, it produces memory B cells and plasma cells. Plasma cells are immediately for producing the antibodies that are specific against this virus or these viral peptides or this viral vaccine that was introduced into the body. The memory B cells are responsible for future infection once someone gets now into encounter with this particular virus whose vaccine was given. So that is the humoral arm. The humoral arm will lead to the production of memory B cells, production of antibodies, and also not shown here, but production of memory T helper cells. So the memory T helper cells and the memory B cells are responsible for future infections, and that is the basis of vaccine. In that you're given that vaccine so that in future, when you get an encounter with that particular pathogen, then you have antibodies in place to protect you. Then on the cell-mediated arm, the dendritic cell, once it's infected by this live attenuated virus, it presents the viral peptides on MHC class 1. So when it's presenting these viral peptides on MHC class 1, it activates a naive CD8 T cell. So this naive CD8 T cells is activated to differentiate into either CD8 effector T cell or CD8 memory T cell. So the CD8 memory T cell is essential for later when an infection is introduced into the body, it fights the infection. And the CD8 effector cells is responsible for fighting that infection there and then via inducing apoptosis of the infected cell. So I hope that makes sense. And both of these arms only occurs when when the vaccine is given to someone that is live attenuated vaccine. It differs, different arms are induced, differs depending on the type of vaccines, and we shall see as we proceed. So as we saw earlier, vaccination induces immunity, and there are other ways that someone can get immunity. So immunity can either be innate immunity that we all know, or adaptive immunity. So under adaptive, we have natural, where under natural immunity, it can be through passive, where you get, the babies get immunized through maternal antibodies, or active natural immunity, where someone is previously infected by that particular virus or pathogen, and from that natural infection, then they get an immunity. In the artificial arm, this is where now the vaccination comes in, where you can be passively vaccinated or actively vaccinated. So passive vaccination is just giving antibodies to someone. So these antibodies have been preformed. For example, if someone was infected with a certain pathogen, then they have formed antibodies against that pathogen. These antibodies from that person can be transferred to another person through the convalescent serum to maybe help in if that other person was potentially exposed to a virus or that pathogen, and you want to treat that scenario there and then. So they can be given these serum antibodies to help neutralize the particular virus. And an example is for HBV, so that is hepatitis B virus, whereby if, let's say, someone is infected by HBV, a healthcare worker gets infected by HBV, and you know that they have been exposed, to neutralize the potential of them, getting further infection, they can be given the convalescent serum from someone who already had been exposed and has high antibody titers for HBV. And then for active immunization, that is when now you give, let's say, a live attenuated virus, or you give a killed virus as a vaccine to induce active immunization for that particular individual. So what are some of the characteristics of an ideal vaccine? I might have mentioned this before, but an ideal vaccine is one that will have a good immune response that is both cell-mediated and both humoral. And I think I'll repeat this. For cell-mediated, the good vaccine or the ideal vaccine should be able to give memory to CD8 T cells. So you should be having CD8 memory T cells. And for the humoral arm, the person should ideally be having memory T helper cells, and also be having memory B cells. So that is, it induces both cell-mediated and humoral of the immune response. Then also an ideal vaccine should give long-term protection, that is ideally lifelong. An example is like the live attenuated vaccines. Most of them are not short-lived. They give a very long duration of protection. Every time you check for the antibody titers in someone who most of the time was vaccinated using a live attenuated virus, the antibody titers are usually very high. And a high antibody titer is directly an indication of more protection. And then again, the vaccine should ideally be safe. So it should not be causing infection, but it should be saving someone from the infection. So it should also not be causing adverse reactions or causing some hypersensitivities. Should just be safe. There might be reactions that are expected. For example, if you introduce something foreign to the body, there are reactions that are expected, but they shouldn't be adverse. And then they should be stable. The vaccine should be stable. So stability can be in terms of if, for example, if it is a live attenuated vaccine, it should not be able to potentially revert to the wild type that can cause an infection. So it should be stable at its state and not cause an infection, like it should not revert to the virulent type. Again, stability can be looked into through the arm of its maintenance and its storage. Some vaccines, they need some very enhanced storage conditions that are not ideal for some environments. For example, if let's say a vaccine needs to be stored at -80 degrees, that will not be very ideal for some places or some countries that do not have that particular infrastructure everywhere to, store that vaccine so that it can be viable. So its stability should be somewhere that can be maintained with even the poorest of the poorest areas. And then in terms of expense, the vaccine should ideally be affordable to even the low and medium income countries. So if it is too expensive, then it does not serve the purpose because it's now concentrated only to those people who can afford it. So traditionally, these are the types of vaccines that have been in place. We have the live attenuated vaccines, the killed vaccines or the inactivated vaccines, then subunit vaccines and the toxoid vaccines. So toxoid vaccines are mainly from bacteria because from our bacteriology, we know some of the pathogenesis of bacteria in our bodies through the toxins that they produce, either the endotoxins or the exotoxins. And then other type of vaccines that have been in place recently are the DNA vaccines and the mRNA vaccines. So we will tackle this one by one, try to understand how they confer immunity, which type of immunity they confer, their advantages and disadvantages. So live vaccines essentially come from whole organisms that are live pathogens, and they have been attenuated, meaning their virulence has been reduced. So they can be, there are various ways of reducing the virulence of these particular pathogens. And then now these pathogens can now be used as a source of vaccine. The advantage of live vaccines is that they can be able to, because they can be able to replicate in the cells in the body, they will be able to induce both humoral response and induce both cell-mediated response, as I said before. So most of the vaccines that are live attenuated, you'll have both memory B cells there, you'll have both memory T helper cells, and you'll also have memory cytotoxic T cells. So examples of some of these vaccines include the rotavirus for viruses. Then we have MMR, which is basically measles, mumps, and rubella. Other examples for for the live vaccines, you can read more about, because I just put in a few here, but you can read more about the OPV, that is the Sabin for polio, then the intranasal influenza, and the yellow fever. So those are some of the examples of the live attenuated vaccines. So as I had said, in these vaccines, the organism replicates in the host and induces an immune response that will cross-react with the antigens of the virulent organism. So this is giving more examples of the live attenuated vaccines, such as the smallpox is also a live attenuated vaccine. But I just wanted to talk a little bit about the disadvantage of this particular using a live attenuated vaccine. First of all, it is contraindicated in people who have immunodeficiency. So if someone is immunosuppressed, they cannot be given this particular virus, or if they cannot be given this vaccine, or if someone is pregnant, they can also not be given this particular vaccine, because there's usually a potential of this particular vaccine reverting. It's less, but there's a potential of it reverting to the wild type and causing an infection. So this shows how ideally the virulence of the vaccine is reduced, because normally the pathogenic virus will be isolated from a patient, and then it will be grown in human cultured cells. So for example, if it was, let's say, a rotavirus, the rotavirus will be isolated from the human, then grown into human cultured cells. Then in the subsequent growth of the virus, now it will be made to infect monkey cells. So once it infects monkey cells, the virus will, when it's in monkey cells, it acquires mutations that will allow it to grow well in monkey cells. Remember, it's a human virus that normally infects humans. So when you use it to infect monkey cells, it needs to adapt to that environment, meaning it needs to acquire some mutations that will allow it to grow well in that environment. So now, once it has mutated, it no longer grows well now on human cells, and that is a good candidate for a vaccine that can be used as a live attenuated vaccine. So this is an example of how you can attenuate a virus and use it as a vaccine. So the second type of viruses is the killed viruses, or the inactivated viruses. And these viruses are also whole organisms. They are killed pathogens, and they can be killed using chemicals such as formaldehyde or heat. Normally, these viruses, they induce only the humoral response, but they do not induce the cell-mediated response, because they are not alive, they are dead, so they cannot actively replicate inside the cells of the, let's say, the dendritic cell, as we had seen earlier. It cannot replicate there. So what the dendritic cell can ideally just do is to phagocytosis and to present it on MHC class 2 to T helper cells, and then it now takes on to the line of the humoral response. So these vaccines, because it is a dead vaccine, it's a dead virus in the vaccine, normally the person would need booster shots. That is the downfall of this particular vaccine. And examples include the polio, again comes here, but a different type, which is the IPV, the Salk. Then we have the hepatitis A and the rabies vaccines. So as I have said, generally, the pathogens will only induce humoral immune response. So there will be no cell-mediated. So on to subunit vaccines. So in subunit vaccines now, we are not using the whole organism. We are using a subsection of the organism or the antigenic sites of the virus that are usually known to induce an immune response in the body. So subunits can be either, for example, for viruses, the subunits can be a protein. And in terms of some bacteria, the subunit can usually be a polysaccharide. So given an example of viruses, you'll have hepatitis B and HPV. So for hepatitis B, the subunit will be the hepatitis B surface antigen. So that's Hep B surface antigen has to be made. And normally they use the recombinant DNA technology to make these particular antigens. So remember, it's a protein. So it's easier for it to induce an humoral response. And then for human papillomavirus, normally they use the core proteins for, I believe we learned about the genotypes for HPV. So they usually use the core protein for the different genotypes like 6, 11, 18, those specific genotypes that are notorious for causing infections with HPV. So for these particular subunit vaccines, they need either an adjuvant or a protein conjugate. For example, if you have a polysaccharide as your subunit, and a polysaccharide will be ideally in bacteria that are encapsulated, such as the strep pneumo or Haemophilus influenza type B or N meningitidis. Those ones, they usually have the polysaccharide subunit. That particular polysaccharide, for it to induce an immune response or induce the humoral arm of the immune response. Remember, the antigen presenting cells can only present proteins. So it cannot present this particular polysaccharide, but this polysaccharide is what is the pathogenic, the virulent factor for this particular bacteria. So in that case, for an immune response to be mounted, then this polysaccharide has to be conjugated to a certain protein. Then when it's conjugated to that particular protein, then the dendritic cell or the APC, the antigen presenting cell that is involved, can be able to present this and this particular antigen to the T helper cells and then induce that arm of the immune response. While for viruses, it's easier. They will not need a conjugate protein because the viruses are already, the viral particles are already protein in nature. For example, the hepatitis B surface antigen is already protein in nature, and also the core protein for the HPVs are already protein in nature. So they will not need that conjugate. But at the end of the day, these subunit vaccines, they still need an adjuvant, because an adjuvant will be essential in mounting a huge immune response. Otherwise, if an adjuvant is not included, then it becomes a little bit difficult for a huge immune response to be mounted and increased antibody titer. I mean, a lot of antibody titers to be available. So all these aspects make the subunit vaccine a little bit expensive, because as we have seen, to even make the antigens, we need to use recombinant DNA technology. At the same time, you still need booster shots for these particular vaccines. So that makes them a little bit not desirable as compared to other types of vaccines. So we also have the toxoid vaccines, and these ones are essentially for bacteria. We all know bacteria, some pathogenesis mechanisms of bacteria is through the toxins that they produce in the body. But in this case, we are just going to concentrate on the exotoxins, because exotoxins are protein in nature. Endotoxins are like the LPS, the lipopolysaccharides that are in gram negatives. So those ones, as we have said, lipopolysaccharides, their potential to induce the immune system is a little bit difficult. So with the protein ones, it's a little bit easier. So these exotoxins are weakened, and the weakened form are called the toxoids, and the unweakened form is the endotoxin, or the toxins that are not good for us. They are ideally pathogenic. So the toxoids are the ones that are taken. And examples of vaccines of this type of vaccines are the tetanus and the diphtheria vaccines. So these are essentially just for bacteria. We don't know a virus that uses the toxoid vaccines. So again, we have the DNA vaccines, which are more presently experimental. The good thing is that they evoke both humoral and cell-mediated immunity. And in the next slide, we'll see an example of how these DNA vaccines are made. But generally, the gene of the antigen determinant of the antigen of interest is usually inserted into a plasmid, and a plasmid is just a circular DNA that is independent. It replicates on its own. It doesn't replicate in the nucleus, it replicates in the cytoplasm. And these are usually mostly found in bacteria. So now, after you've identified this gene of interest of that antigen, you insert it into this particular plasmid. And then the plasmid is transformed into other bacteria, so that now they can make more and more and more of that particular gene. And then this gene, is injected into the host. Maybe if you're trying to, if your host at that moment is, let's say, a mice, you inject it into the mice, and then the foreign gene will be expressed in the mice. It will be transcribed, translated, and then sufficient amount, after translation, sufficient amount of foreign proteins are produced. And these will now elicit, and remember, your foreign proteins are the antigens of interest, which will now elicit an immune response. So it's currently being tried for hepatitis B and HIV. So these are still experimental. So this is what I was talking about. You have your gene of interest that codes for the antigen, which is also an immunogen. And then you transfect it into the plasmid. So the plasmid now is transformed to more bacteria, and the bacteria will be grown. And we have more and more bacteria. You purify your plasmid DNA, and now you inject it into your host of interest. Now you'll, once it's injected, after transcription, translation, the proteins are produced, and they are expressed, and you see if your immune system or the mouse immune system is producing antibodies against this particular antigen. So this slide gives a classification of what we have been talking about, and it gives a short description of, the types of the vaccines. So you can categorize them into the whole, agent vaccine, or subunits. Under the whole vaccine, we have the inactivated or killed, and the attenuated, And example for attenuated, and we remember what we said about attenuated, live attenuated, they elicit both humoral and cell-mediated response, meaning there is memory B cells, memory T helper cells, and memory cytotoxic T cells. Then examples include MMR, and an example for viruses, you can include OPV and intranasal for influenza there. And then for the inactivated or the killed one, remember we said this one you will need booster shots, and it elicits only the humoral arm of the immune response and not cell-mediated. And an example is the rabies vaccine. For subunit, we've talked about the different types of subunit, the polysaccharide and the protein, and we've seen that the protein are mostly for viruses and polysaccharide for bacteria. And we've also seen that the process of getting these particular antigens can be a bit expensive. And with these, you will also need booster shots, and to have a bigger, to mount a huge immune response, you will also require adjuvants. And an example is hepatitis B vaccine. We saw HBV, and for bacteria, you remember there was strep pneumo and meningitis. So what are some of the advantages and disadvantages for these different types of vaccine? So, for example, for the attenuated vaccines, the good thing with them is they will closely resemble the natural infection, and thus it gives an excellent immune response because we get both cell-mediated and humoral. And then you don't need adjuvants, you don't need booster shots, and it gives you long-term immunity. The disadvantage is that there's a potential of reversion to the virulence type, and also you can't give it to immunocompromised individuals and pregnant women, and also it's less stable, and you ideally need to have it under cold chain. For the killed vaccines, they are a little bit safer, and they are more stable in adverse environmental conditions. The disadvantage is that the immune response may not be long-lived, therefore you need booster shots for the same. And you may also need more than one dose. For example, you get one dose of the vaccine, and then after a certain period of time, you're given another dose. Apart from needing a booster dose, you may have to do more than one dose. And then they are more costly also. The process of making them is costly. And the subunit vaccines, also the advantage is that they cannot cause disease. They are safer than live vaccines, and they are more stable. The disadvantage is that their effectiveness is not guaranteed. It's not guaranteed that it's going to mount an immune response. And if it does, then the immune response can be short-lasting. And sometimes people can have hypersensitivity to the adjuvants that are used in the subunit vaccines. So normally they're undertaken, mostly they're used during epidemics, not all the time. So toxoid vaccines also, they don't cause disease. Disadvantage is that they are not highly immunogenic like the subunit vaccines, and they may also require multiple doses. With DNA vaccines, the advantage is that it can induce both humoral and cell-mediated. It is stable, you don't need cold chain, and it can be designed via genetic engineering to have your desired antigen the way you want your antigen to be. You can engineer it using DNA technology. But the disadvantage is there's the question of safety, because the problem with inducing foreign DNA into the body is that you don't know what it will do to the DNA in the body. It might cause instability in the DNA. So there is a question of safety. And also efficacy, generation of DNA plasmids. It might elicit low levels of T and B cell memory. Uptake of those plasmids is where the efficacy is being questioned, because sometimes you might end up, those plasmids with the DNA of interest might end up not being uptaken. So generally, this slide gives advantages and some of the disadvantages of vaccinations in general. So what are some of the advantages of, taking vaccines? Ideally, vaccines, they are able to protect people from diseases. They are able to eradicate epidemics, such as smallpox has been eradicated. They're able to prevent spread of diseases to others. So if it's a vaccine that can confer herd immunity, then it can also protect those people who are not vaccinated. Then it prevents the potential greater cost of treating infected individuals. The disadvantage is that it's not guaranteed to work 100%. There are other cases, questions about safety, questions about its effectiveness and efficacy. The possible side effects based on what has been used to make the vaccines, for example, the adjuvants, and sometimes constant booster injections are usually inconvenient to people. People don't want to be injected all the time. So some of the limitations, which is maybe just a repetition of the last slide, include for vaccines include reactions to inoculation, reactions due to faulty techniques, hypersensitivities, and then some studies showing that some vaccines are associated with some of these syndromes, such as the GBS and encephalitis, for example, in rabies and smallpox, and death, and so many controversies surrounding vaccines. So those are some of the limitations in vaccinology. So this particular slide just gives an outlook of how vaccine development takes place and the different stages in vaccine development. So for example, we have the preclinical stage and the clinical phase. And under the preclinical phase, we have lab studies where essentially you're doing studies about the infectious agent. You're doing studies about the infectious agent. You're studying the epidemiology of that particular disease. And you're also trying to understand that particular antigen. You're just doing antigenic research. So these are lab studies. So you try to understand how that antigen in that microorganism looks like. So you're trying to, ideally, you're trying to generate a potential vaccine. Then in animal studies now, this vaccine that you have potentially generated in the lab, you give it to the animals. So in here, you're trying to study the immunogenicity of the vaccine. You're trying to study how safe the vaccine is. And also you're trying to just know if the efficacy also of the vaccine, you can also tell at this stage. So once you give the animals that vaccine, then you expose them to that particular virus, and then you see if that vaccine has protected the particular animal that was, for example, if it's a mice, if that vaccine protected it. So you'll see at this stage if the vaccine is safe, if the vaccine mounts an immune response, and if the vaccine has some efficacy. Then now when you go to the human trials, the human trials have 3 phases: phase 1, phase 2, and phase 3. In phase 1, you're ideally dealing with a small population of people, around less than 100. And normally these are healthy individuals with no underlying conditions. So in this stage, you're trying to understand, again, the safety of the vaccine. You're also trying to understand the immunogenicity of the vaccine. And also you're trying to understand the dose that you can give that will work within this, within the population or within this particular population. So now when you come to phase 2, safety still cuts across throughout. But remember, in phase 2 now, the number of individuals you're dealing with increases to up to 500 or even up to 1,000 people. And also in phase 2 now, you're not dealing with healthy individuals. You match your population now. So your population should include all types of people, both healthy, both people with underlying conditions, different ages, different races. So here you will try to assess again the safety. You'll try to assess the optimal dose that you can use for this particular vaccine. And also you try to assess the effectiveness of this vaccine. Then on phase 3 now, the number of people goes to thousands. And also this, the population is inclusive of everyone. Like it cuts across children, adults, different races, different people who are also healthy are in there, people with underlying conditions are in there. Then they are given this vaccine and they are in the population. They go on with their daily activities. They are not confined somewhere. So here again, you're just trying to assess the efficacy, the effectiveness, and the safety in a larger population. And then now you do your market research and sell your vaccine as a biotech company, maybe, probably. So these stages take a long time, all the way from preclinical studies to human studies. Normally, they will take a very long time, except when in cases of pandemics, where now some of these stages get a little bit rushed for emergency use authorization. As we saw with COVID, the vaccine development took a little bit of a short time because of emergency use. And those are usually, FDA now approves them, but it's emergency authorization. So these are some of the recent advances. We've talked about the DNA vaccine, and we've seen that it's experimental. Another recent advance is the mRNA vaccines, which we have, most of us, or not, if not all of us, have heard about, especially during the COVID era. And I've attached a video here, some YouTube video on mRNA vaccines that there's a guy that explains the different types of vaccines very well, their mechanism of actions. I don't know if we have time, I can try and, put the video up for us to see. And maybe if you guys are interested, we can watch it together and learn together some of the mechanisms. But if not, then you can go ahead and check it out for yourselves, and then you let me know what you think about it. So that's all for the presentation today. Please let me know if we can put up the video. All right. So let me try and I'll have to stop sharing and try and put up the video for us to.
About 3 vaccines that.
So Kelsey, can you tell me if you can hear the sound of the video, please?
Being funded by Operation Warp Speed. We're going to talk about AstraZeneca,
Yes,
or the Oxford
you can hear
vaccine,
it.
Moderna.
Is it okay? Can you hear the sound?
Yes.
All right. So I'll put it up. It's a little bit long. This guy explains something that I found a little bit interesting, but I'll put it for like maybe 15 minutes. We'll see, and then we'll discuss. So let's have a look and concentrate.
Let's go ahead and get started. All right, so what we're going to do is we're going to talk about vaccines, again, the ones that we're talking about that are funded by Operation Warp Speed, Moderna, Pfizer, BioNTech, and AstraZeneca. What are the phases or stages that they have to go through to receive FDA approval, in this case, emergency use authorization? So what we have to do is first have a complete understanding of what the goals of vaccine development is. So throughout all of these stages, there's two primary.
Dr. Obiero,
Yes.
the video is frozen from our end.
Oh, okay. Let me start it. Is it okay?
Okay, from my end, it's still frozen.
It's still frozen. All right, let me
What's up, Ninja Nerds? In this video today,
let me stop sharing and try again.
we're going to talk about COVID-19 vaccines. We're going to
How
primarily
about
talk
now?
about 3 vaccines that are being funded by Operation Warp Speed. We're going to talk about AstraZeneca, or the Oxford
Oh
vaccine,
yeah, now it's
Moderna,
moving.
and then Pfizer,
Some students have
BioNTech.
asked if it could be in
What I
full
want
screen.
to preface.
All right, okay. It's in full screen.
So
Is it okay?
Start this video talking about how these vaccines are being developed. What are the phases and stages they have to go through? Then we'll talk about the mechanism of action, how these vaccines.
Okay, it's frozen. It's frozen again.
And then
Oh.
finally, we'll go over some of the.
I think what we'll do, because once I put it in full screen, it just freezes. So I don't really know why that's happening. So maybe you guys will just watch it on your end.
Yeah, that's okay.
Yeah, maybe you'll watch it on your end. I've just put the link there. So someone is saying that, do you want me to put the link on the group? All right, I can do that. Let me put the link on the group so that I'll put it also here on chat. Okay, so maybe we can now tackle the questions. The moment I stopped sharing, all the questions on chat, I think, went. So if you don't mind, but I'll try to scroll and see. Okay, so someone is asking if the class is until 7 p.m. No, we will probably finish in the next 30, so don't worry. So CD8 effector doesn't have memory in case of the same infection. So I don't know if you're asking based on live attenuated vaccines, but remember what I said about live attenuated vaccines, they elicit both cell-mediated and both humoral. So when we say both cell-mediated and humoral, under the humoral arm, we will have memory cells for B cells. There will be memory cells for T helper cells, that is for the humoral arm. Then on the cell-mediated arm, we have the CD8 cells, right? So the CD8 cells will also have, there will also be memory CD8 T cells. That is the case for live attenuated. In the case for the inactivated vaccines, remember we only said they only elicit the humoral arm of the immune response. So we'll only have T helper cells and B cells memory, but no CD8 memory because cell-mediated is not there. Okay. So what did she say about live attenuated and pregnant women? I said they are contraindicated for pregnant women and immunosuppressed individuals. And someone has answered that. They can't be given to pregnant women and immunocompromised people. Remember the embryonated egg, the membranes connect. Okay. Since it has the potential of reactivation. Okay. Do live attenuated viruses require boosters? No, they don't. Remember, one of the advantage was that it is most of the time it's lifelong. It gives a very long immunity. About 15, 20 years is when you might need booster, but sometimes you're told, like, for example, if you've taken the shot for the yellow fever, I think they tell you to go back after how many years? So many years, it's usually 10 or 15. So for a booster shot, but most of the time it's not even required because most of the time it gives a lifelong immunity. Okay, IPV or OPV in kill vaccine. So OPV is oral polio vaccine, which is the Sabin. This is the oral one, and it is the live attenuated one. You need to differentiate it from IPV, which is intramuscular, and it is called the Salk, which is the killed one. I hope we are together there. I can see some responses. So the idea in the chicken embryo cells being from a different species cause mutations in the virus that makes it unable to be virulent in humans, but it can revert at Harrison. I don't know where this one is stemming from. Maybe if you can, Kamunya, please put your question so that we understand well what we are talking about. I'm thinking you're talking about the process of attenuating the virus, right? So we'll come back to that one. Okay, in Hep B, between the serum induced and recombinant one, which one is better? So with passive immunization is usually like a short term, I'm getting you at that point, like I've been exposed at this particular point. I need to neutralize this particular virus before it goes and keeps and infects other, other cells. So it's a moment of, I'm neutralizing the virus at that point because I've been exposed. But with the recombinant one, remember, it's going to through the whole adaptive arm of the immune response. And it takes time, right? Adaptive arm of the immune response is not immediate. So it takes time. So if in case someone was exposed and you give them the recombinant one, it might not be beneficial at that moment. This one, especially a better description for passive immunization is with a bacteria called tetanus. So if someone is exposed to tetanus, normally they are given the serum antibodies, but at the same time, they will also be immunized at a different site, because you can't give the vaccine and the antibodies at the same site, because the antibodies is just going to neutralize that vaccine there. So one site, you're given the serum that has the antibodies to go and neutralize the bacterial toxin that was already in your body. And then on another site now, you're injected by now the vaccine itself. So that's a better explanation for passive immunity. But the idea of passive immunity is just to get the virus or the bacteria there and then, so that it does not continue replicating and infecting cells, because the other one, the recombinant one, will take time to develop. Okay, so someone is asking COVID vaccine, what type? So the video that I've given you is going to describe those vaccines very well, but maybe I can give you a glimpse of which different ones are which different types. So we have the Moderna and Pfizer, which are mRNA vaccines. Then we have the AstraZeneca, which is more of a DNA vaccine that uses a viral vector together with Johnson & Johnson. So you can go and read more about them, how mRNA vaccines work. We have seen how DNA vaccines work. So if you want to understand more, you can read about how mRNA vaccines work. COVID siko na more study kina subunit. Yeah, COVID has so many. So sometimes what is used in most countries, you can concentrate on that, on understanding those. So Johnson and AstraZeneca were viral vector vaccines. Yes, Mohammed, that's correct. Pfizer and Moderna are mRNA. That's correct. Okay, I can see most people agreeing. Someone has written 43. I don't know what 43 means. Or 43 minutes. Yeah, go and listen to Ninja Nerd. He has some very good explanation there. I put the link there about someone asking about the link. Okay, so what was his name? Someone asked about because I can't see any other question. Maybe we can tackle the question on embryonated egg. I can't remember the name of the person, so I'm just scrolling through to see. So, Kamunya, would you want to write it down again so that we talk about it? You say you are answering Harrison. So what was Harrison's question? Harrison, do you want to type so that we tackle it and and close up. Okay, what does it mean, a vaccine being virus-like particle? I bet you're talking about, because for the vaccines that they're calling virus-like particles are usually, most of the time, the DNA vaccines. For example, usually it's the adenovirus that is usually used. So this adenovirus acts like a virus receptor. And then this adenovirus, you put the antigen of the vaccine of the particular pathogen that you're interested in the DNA of the adenovirus. So then you make the adenovirus infect other cells. For example, if it becomes the vaccine, you use it to infect the cells, and the DNA of the adenovirus is going to express that. Remember now, the DNA of the adenovirus has is coding for the antigen that you want, that is of interest. And then it will produce antibodies that will act as antigen. So that's what I would call a virus particle kind of vaccine. But the virus-like vaccine that you're talking about, I guess it's the HPV vaccine. I would say HPV vaccine is more of a subunit, because it's coding for the core proteins. Maybe they're calling it virus-like because you remember virus has the core protein and the outer protein. So the HPV vaccines has the core proteins, or the core proteins for HPV. For example, the HPV of interest, like 6, maybe 16, 18, 11. And it has all those core protein of that of that particular HPV. So it acts like it's the virus, but it doesn't have the outer envelope. Remember, it's not a whole viral particle. It looks like a virus because it has the core protein, if I think of it from that sense, but it is not the whole virus. It's a section of the virus. That's why it falls under the subunit still. So Kamunya is saying that he was claiming that contraindication for live vaccines in pregnant had something to do with them being cultured in embryonated eggs. Okay, no, the answer is that it's contraindicated in pregnant women because of the potential of it, reverting to the wild type and causing an infection. That's why it's contraindicated in, you don't want, because ideally the pregnant women are most of the time always categorized as slightly immunosuppressed, if I could say so. So if this one reverts, it can cause more damage than good. Is it okay now? Okay. So are we all together? Can we close the day? So all right, we can close from there. Kindly go watch that video. It will be very informative for you. It will give you more knowledge on mRNA vaccines and DNA vaccines. So and I hope you get something out of it.
Thank you.
Thank you very much, and enjoy your evening.